Okay, so I talked about one way of predicting subcellular localization by using 20 input units that code for the amino acid composition of that protein, so what fraction of that protein is adenine and so forth, to ultimately classify that this protein is in localization L or not. To use the surface composition and to use sequence and, for instance, secondary structure. And then there could be a second level that does localization to localization. Essentially, it has these two output from the previous layer as input and somehow sees commonalities in the prediction. It's like simple averaging. So the simplest way of this second level here would be an averaging. So saying whatever in the first level here is the highest value is taken. I take the winner. Right? Winner meaning I take whatever difference here is the highest, and that is in fact the one that I choose, or whatever is the highest value. There are two ways of doing that. That's the simplest way to implement this step. On a more complicated step, you have a new machine learning that essentially does this. Now in the new world of deep learning, you in fact would learn the whole thing as one step and would have this as intermediate layers, right? In this way we did it as two different devices that are trained with the same data set. Same data set meaning in the same partition of training and testing. And then ultimately we have the issue of the classification in the end, where again this is L or not L. Now, then the other way of doing it, or not the other way, so the way of bringing L's and non-L's together could be the classification, whatever is the highest value. It could be a sum over the values, and you can also put it into some pathway or onto a trafficking pathway such as the one in the cell. If we put it onto a trafficking pathway that is hierarchical in a parallel architecture, then we would have the advantage that for some of these decisions, for instance, does the protein go to the secretory pathway? We know for that we need a single peptide. So my classification device that says secreted or not could use different input information than the one that does nuclear or not, right? That's an advantage. So here's one solution in doing this, where I show SVMs that do two binary classifications of, on the first step here, secretory versus intracellular, and so forth. So they go further down the line. So all of these nodes here are differently learned SVMs, for which I in fact have the freedom to choose different input in order to sort of map the problem. The first issue that I have asked is, how would you choose the architecture, the tree? So which would you put on the top of the tree, or the root, or whatever you say? So what's the first decision? How would you choose? In this particular case, you see the choice, secretory versus intracellular. But in principle, you could have any choice there. And how would you choose which one to choose?
Maybe the two compartments that are easiest to...
What's the advantage of that? So essentially you begin with a choice with that particular solution, with that particular decision that is easiest to do. So for which, to translate this statement, for which you have the highest performance. What would be good about that?
You don't have to do it later on for each of the different nodes?
I'm not entirely sure that I understand what you're saying, because you still have, the architecture does not change.
You don't have, at the lowest level, you don't have to do the same check like for each, like if you don't do intracellular and secretory pathway, if you do membrane and non-membrane, you might still have to check for all non-membrane proteins if there's secretory pathway anyway.
Yes, but Johannes, you're now making a very generic observation. Whatever you put on top, you will not have to do further down. That's all you say.
Yeah, but
and on that level, it's true.
But if you have a more general decision at the top, you don't have to make it in multiple nodes at the bottom.
But in fact, you go a step beyond and you assume something. So you actually now talk about something that is more significant. You talk about more, she did not say for most points. That is a decision that is valid for most points. She said a decision that is for which I'm doing best, which could be a minority decision. It could be that this is in fact classifying. Let's just do. Let me do a dumb thing. Chloroplast. So I want to predict the human protein, and the simplest decision is chloroplast. Without training machine learning device, I will always say no. Okay, so this would not be the kind of thing that is generic at all. You will never have to do the chloroplast decision again.
But that's a very specific decision and not general, like I said.
Yes, but all she said is the most accurate. So I believe what you did is you read the labels, and in these labels your answer is right. It turns out that this is a more generic decision. And you're right about this. You may have to do that classification further downstream anyway, because it's one that will ultimately happen. So let me just rephrase what you say as a proposal to say, you do up there, you do the decision that splits most points. So that is not necessarily the most accurate one, but it's the one that achieves the most classifications in some sense, right? But I'm still asking, what's the advantage of doing the most accurate one? Yes?
Shouldn't it reduce the error further downstream?
Why?
Well, if we start with the decision where we are most sure about, then this is by definition the decision that is the least likely to be false.
Yes, there's one way of looking at it. It's a good way of looking at it. And there's an additional, yeah?
And also something just popped into my mind. Maybe it could be the case that some of the classes that are on different branches have a similar support vector profile. So if you first sort them into secretary and intracellular, then you might, and if that is accurate, and then further down the line you have, for instance, let's say organelle and nucleus have a similar vector profile. Then if you split it before, you also kind of catch that error beforehand.
So, but that would, so you actually advertise that may be an added benefit, but if you pulled your argument through, then you would have to look at the overlap, the profile overlap.
Okay.
And then classify according to the profile overlap. I believe what you're recommending here is option 3. So we would have accuracy, number of decisions made, and then sort of overlap or similarity between the points. I believe this is a third way. This is a third possible decision. But let's get back to the first point, which is the accuracy-based one. Anton says that the probability of it increasing the overall accuracy is high because you begin with the right decision. But another important, and that is right, and there's another important aspect, which is if on that decision you made a mistake, you went this path by mistake, you will classify. Whatever you do afterwards will be a mistake simply because you made a mistake up here, and you cannot correct for that. So ultimately, that is the other reason. So you reduce the number of mistakes by misclassifying in the first level, right? And this is maybe exactly not along the line of what is the most common separation. Yes?
Well, my assumption was that the classification was to be done anyway, not at the first decision, but at a later on. So
but if it is a complicated one, even if it's the most important one, this would argue that if what you're talking about is one that is coming again and again and classifies many points, but you're very poor at doing it, then actually you want to sort of rather do it 5 times down here than getting it wrong once up here.
Not like if both the first and the second one are the general ones, like intracellular and secretory and membrane, non-membrane, I guess, then it wouldn't really matter which one's the first and which one's the second, I guess.
Actually, it does matter. So on this particular example, it does matter. I don't know why you picked them. You know about log tree 2. So in log tree, we did it here. In log tree 2, we included the membrane, and actually made a big difference which one was first. And both of those were much better than any other classification. But anyway, so one aspect really is the beauty of this is you can, at every single node, decide what your input is. This one here we know is the secretary pathway, so we know the internal composition is irrelevant. Now, here's another aspect of this separation, that down here, it will not use all the data. If we go along the intracellular path here, this SVM will never see secreted proteins. So the training set will also change. So yes, you change the way the input is, but in the logic, and again, you can try it in two different ways. You can show all the proteins, or you can show those. And we tried this 2 times, in log 3 and in log 3.2. And in both cases, again, I'm not saying this is always going to be like that, but in both cases, it was much better to have a decision here that is specific to what happens at that point in the training, even if I made a mistake. So bringing in the other problems, the secretary proteins from here, back here, because I can make prediction mistakes, did not improve. Okay? Again, this may not be always true. Now, another thing that you have to realize is that the further down you go, the more difficult typically the problem gets. Well, this is sort of the same thing again, right? If you make the split of the tree such that the simplest or the most accurate classification is up there, then down here they will get more difficult. But it's also true because you are, in the logic, you're trying to make decisions here that are more fine-grained. So the major things between intracellular and secretary is already out in this idea here. And then here, well, mitochondria and cytosol, in fact, is not a good example because that may still be okay, but there are other classifications where we look into subnuclear, and then the story gets much more complicated. So the accuracy drops. Okay, so let's begin with the classification. So on the first level, so what I'm showing here is essentially the ones that are right in a 2-state classification, because every one of them is doing a two-state classification. And then we move further down, and you see the numbers are going, getting lower and lower and lower. And this is ultimately shown in this table here, where we compare a bunch of prediction methods. And I shouldn't spend too much time on this table. It always has accuracy and coverage for different pathways or for different decisions. And you see that ultimately the accuracy goes down. And this is sort of the overall accuracy. And that the method performs very well compared to other things. Again, the pro is you have better discrimination with the tree, and you can sort of adjust the way you do the SVMs to the problem. And the problem is really that you cannot recover from mistakes you made on a higher level. And there's just no way out. You can try to hack around it by sort of summing again over the tree, but it turns out, so essentially, if you try to recover from this problem, then it means that at a lower decision you no longer believe what happened up there, and that essentially means you get rid of the tree. And we are back to the pro. The pro is the tree works. So that's why this one doesn't work. The hack doesn't work. At least all the ways we tried to do the hack, and it's convincing. Now it turns out that the tree that we chose is somehow reflecting what we see in biology. In some sense, that is sort of what you meant, I guess, Johannes. The most relevant decisions are somewhere up there, and they happen to be the ones that are relatively easy to do. When we look at this entire proteome annotations, then we see that yeast, Arabidopsis, and human overall have some similarities. What I should have put in here is the manuscript we are currently writing up. So now you can predict the subcellular localization for the entire organism in 7 states, and you can define a vector, and then you can measure the difference in the spectrum of subcellular localization between different organisms, and you can essentially take this difference and classify them in a tree. And it turns out that although the differences in subcellular localization are relatively minor, this classification reflects biology. So it does really work in that sense. But that is much later in this. Oh, well, maybe I could actually, that is an interesting point. Let's turn that into a little session of a whiteboard session. And let's just look at a simple issue here, namely the issue of the extracellular proteins. Say you had a classification device that predicts whether the protein is extracellular or not. That's all we do. And we want to identify the extracellular proteins because those are the ones with which bacteria try to attack eukaryotic cells, for instance. That is one reason why you could be interested in that. And you, for instance, may want to look for bacteria that have the highest fraction of secreted proteins because you may believe that those are most pathogenistic. So they're most aggressive. Now, again, my device has, say, 8% extracellular and correspondingly 92% for one bacterium. Okay? So this is the non-extracellular state, non-X. And now I run this device on 20 different organisms. And what I'm going to compare these organisms, I want to sort of classify them according to their the degree of extracellular things. I want to establish what's the spectrum of things they use, or let's stay here in a eukaryotic example, where we have in fact many states of localization, right? And I want to classify them according to the similarity in these states. So we throw chloroplast in that situation, we would throw chloroplast out, we would recount this pi, we would only include things for which we can completely compare those three. So that comparison shown here is not quite right. So my question to you is, would you take the prediction? And let me just rephrase this, that's where for the first time we sort of encountered this issue. We have proteins that predict transmembrane helices, and in human, they predict that TMH, so proteins or TMP, proteins with transmembrane helices, in human, these are roughly 25%, some people say, and 75% are not TMP. So we have 25% of the proteins in human, are roughly 5,000, have transmembrane helices. Okay, that's what the prediction method does. Is that the way you would estimate the number of transmembrane proteins in human? So obviously, the correct answer is no. The answer may be yes. But I try to induce in you that the future answer will be no. In fact, this is what 100% of all the publications that we looked at for this particular issue of transmembrane prediction have done. So it's fine if you do that too. You would align yourself completely with what has been published. And it's still wrong. Why is it wrong?
Well, it's on a gene basis and not splicing.
Oh, that actually is a debatable one. Johannes, that point is taken, and let's not get into it because we don't actually quite know which direction this falls. So you could argue both ways. This is a very interesting one. You could argue.
One is you don't know about the expression, so it might have only a few genes or different proteins, but it doesn't say how active it is in that regard. Or like
yes, and this is also, it's also not cell-based. So this is assuming that every single protein in human is expressed in every single cell, which most likely is not the case. So it's also wrong in that one. That's true. But the mistake I'm talking about is on a different level. Because what you say are, once I put in my correction, your attacks will still be valid, unfortunately. But my correction helps a lot. But what is wrong in this?
I don't know, but I can take an educated guess. Maybe.
What is the education of? Sorry.
Maybe it's because they are it's transmembrane proteins.
You could.
I have nothing to say. I have nothing to say.
Then would you have something to say if we went back to the X and not X, where X is extracellular? Would that change?
No, because it's like transmembrane proteins. I was thinking you meant only membrane proteins, and then it's like a question of what is a membrane protein and what is not, and yada yada yada. But that goes basically in the direction of what Johannes said with the per gene basis.
Guido, immer noch, nicht Johannes, Guido. But
okay.
To your point, it's actually true because we're looking for transmembrane.
Oh, the name is so the Johannes rest part is so much closer to you. Yeah, it's so much closer to you because you why do you two? I mean, come on, look at how close you stand to each other.
Oh.
Sorry, Guido.
No problem.
Self-defense. Yes, Guido.
if we are only deciding between transmembrane proteins and not transmembrane proteins, we ignore membrane proteins, non-transmembrane spreading, and those have a overlap with membrane, transmembrane, so they're likely overestimating.
This is not quite what I mean. There's something else wrong in this. Something much more basic.
So you
Yes?
said that, so the method identifies one quarter as transmembrane
Yes.
and the others as non-transmembrane.
Yes.
But that doesn't mean that that reflects the distribution within the human proteome.
Right? Why not?
Because
That is correct.
we
Why
know
not?
all the proteins. We have it.
No. So yes, that is also. So you join the club of good arguments against me, but all of those I'm not defending against.
Then the next one is membrane proteins are harder to resolve than non-membrane proteins.
We are getting closer to the point. So actually, say we have a data set of 20k human proteins. So this is where the problem is coming from, but the real problem is somewhere else. The real problem is when you develop a method that predicts membrane proteins. How do you do this? What is the data set that you begin with?
Amino acid sequences.
From what type of proteins?
If you want to know if it's transmembrane proteins, you don't know what kind of proteins.
Usually you begin with a method that predicts the membrane helix. That means usually you begin with a set of membrane proteins or transmembrane helical proteins. And then typically that's exactly what your secretory pathway. Typically you always begin to learn something that is specific to that problem. Identify membrane proteins by identifying membrane helices, and then you see you don't find membrane helices, you call it a non-membrane protein. But actually the reality is that typically methods, good methods, tend to have something like an error here of 1% and here of 5%. Because they are trained.
I have a semantic question.
Yes.
And I apologize for it, but we might want to define this now, because many people, they for instance, when you say membrane protein, what you actually mean is transmembrane protein, right?
That's why I write TMP.
Yeah.
And that's why I say transmembrane protein, correct?
I know. It's I don't want to be a dick about this, but it's
and again, so the problem,
it messes with my head when you say membrane.
that is very, very unhealthy. So transmembrane protein is defined as something where the helix goes through the entire layer, the lipid bilayer, and there's at least one helix that goes through it. When we look at these numbers, roughly ballpark speaking, half of these, so 12% of those have a single membrane helix, and that is very difficult to distinguish from a membrane anchor and from a single peptide. So there are all kinds of issues there, which ultimately pertain to why this error rate is so high. But transmembrane protein is still, at least there's one helix that goes through the entire lipid, so it's not an anchor. Because again, the number of anchor proteins there are a couple of methods out there, but our ability to predict those is so poor at the point, and even our ability to see them. My method has nothing to do with that, and I'm saying TMP. Okay, TMP. Or TMH, TMHMP, blah, 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 blah. Enough now. TMP. So, since the mistake is so different between these two classes, and since this class is the majority class, so you make most of your mistakes happen here. Okay? So let's just assume that in the particular example that we have here, we have 5,000, we assume that really in reality 5 of the 20,000 human proteins were membrane proteins, right? On average, what you will do is you will make 1% mistake in those. For the 5,000 you will make 50 wrong. So for the TMP part, you will have 5,000 minus 50. So you will identify 4,950. Okay? And for the non-TMP part, you will in fact have the 15K minus 5% of 15K. What is that? You guys are young and fit.
750.
750.
So by mistake you mean false positives in this case?
False negatives and not TMP case, right?
Yeah, false negatives, right.
Yes.
It's both false negatives.
So overall, you will make 800 mistakes, and 90% of these of the mistakes are for the non-TMP. So you will call them
Negatives.
you will call them negatives, but they are actually membrane proteins. And you also see that this one here is affecting the mistake here substantially more than the mistakes you make here. So the ones you miss as membrane proteins is more than 10%. Yes, it's more than 10% of the actual membrane proteins. So this mistake on the membrane proteins themselves is not really right. Then you will also see that your ratio, your estimated ratio from the numbers you actually predict will be off. This will not say 25%. This will give you a very different number. And it's relatively easy to correct for that. Right? You only have to pencil in the error rate. When you pencil in the error rate, you can see what on average, you don't know what is the right membrane protein. You will not correct the prediction. But you will correct the prediction for the proportion. Because you know the mistakes. You know the confusion matrix. You know exactly these numbers that I gave you. How many true positives, true negatives, false negatives, and false positives. So those 4 numbers you know for your method. And since you know those 4 numbers, you can correct the estimate of the overall content. And that is the case here. Now you can sort of apply the same logic to a method that has 7 or n different output states. The idea is clear? I take this to mean no. Okay, let's go slow. So everybody is clear about why I said minus 50 and minus 750 here. Since you do know that you're expecting to make that mistake, you essentially can do this one. So you look, well, I'm sorry. Yes, I see I lose you on that one. So let me do it differently. Predicted TMP times 1 plus 1 over 100. No, wait. That is minus, so I have to subtract this 1 over 100, and I have to add the 5 over 100 times predicted not, right? So this will be the estimate, the error-corrected estimate for my prediction of membrane proteins, for the number of membrane proteins. Did I make a mistake?
The parenthesis is too far off. It needs to stop after 100.
Which 1 plus minus?
Or the plus.
Plus?
Because it's the other.
Oh yeah. Oh yes. Yes. Thank you. Yes, thanks. Everybody clear about this?
I'm not, but just go on without me. I'm just going to read up on it later.
Well, you don't need to. This is anyway is a brief interjection. What I mean is you get a number of the ones you predict to be membrane proteins. Sorry for the TMPs. And the number that you predict to be non-TMPs. Okay? You know that in the ones that you predict to be TMPs, 1% is a mistake. So the actual number, your estimate for the actual number is 1 minus 1%. Plus the ones that are predicted as non-membrane proteins, which you also make a mistake on. The mistake on the non-TMP is given by the number of non-TMP times 5%. And that's this number. And that gives you your estimate for the true number of membrane proteins. It gives you an estimate, error-corrected estimate for the number, not for which ones those are. So that doesn't help you to identify which one are the membrane proteins, but it gives you a number. And now you can do the same math for the number of not transmembrane. So should I go through it?
No.
Anyway,
The inverse formula of that.
it's the inverse formula. And then you have the error-corrected ratio, which differs substantially for most prediction methods from what is published, or from what you get directly out of the prediction method. And it turns out that if you do this error correction, you get actually remarkably close to the true distribution for methods that have many mistakes. The next surprising reality is it's not always the most accurate method that estimates the composition best, because a method can be not as accurate but still relatively unbiased by particular classes, while a method could be better by getting the first split right, but then it could create a relatively biased prediction and could become bad at actually estimating this ratio. And how can you know which one it is, whether the best method is better or not? You can only find out by trying it out on different methods. Okay, so let's get into some other prediction methods. So there is a bunch of methods that use amino acid composition, then targeting motifs and amino acid composition, and then essentially only targeting motifs. Let's go one by one. P-sort, that has started with Minoru Kanehisha. Does anybody know Minoru Kanehisha for anything? Oh, actually, sort of this gives it away. KEGG. So this is the man who had the vision to build KEGG. Essentially, maybe you grew up with this sort of thing, building a pathway of proteins. What is a vision in this? But when Minoru started around 19-something, nobody believed that he could achieve this, and it was perceived as utter nonsense. And well, you all live with it. Sort of before he retired, he pushed it to the level where this became relevant for the bedside, so for which it moved from bioinformatics to really implicating clinical research. Bench to bedside is one of his latest messages, and he is a giant in the field. Now the other person was Kenta Nakai behind P-sort, and again it's one of the big names in the field with a tremendous success. The first idea they had was P-sort. The beginning was really that there is a way to predict subcellular localization from amino acid composition, and then there are some motifs. Let's put these two things together in some sort of simple way, and we have P-sort. Then the later point of P-sort 1, so the logic really is, that's the early one, it begins a little bit like that one before. So do you have a signal peptide or not? And the rationale was, because at the time being, that is very much in now, for the change of it, let me call it Yugido. So now this was done initially because it had a lot of classifications. So this tree now is based on your idea. Simply, I have data, and I can do classify many proteins with that. And you see that the transmembrane story comes later here, where again there was data for it, and then there were sort of minor other issues, and most of those were database lookups in the early days. So the protein was known to be there or there, and you simply looked up, is that one of these proteins, or is it similar to one of these proteins? Then added to the group came Paul Horton, and Paul Horton essentially introduced some concepts of machine learning that became then P-sort 2. Ultimately, k-nearest neighbor classifier, and then more generic ways of using machine learning to improve. And again, so there's a bunch of motifs in there, a bunch of different features, DNA binding motifs, and many other motifs. GPI anchors, here we are on the membrane story, the membrane anchors. There's coil structure that sort of gives a preference for binding DNA, and therefore in a very indirect way to sort of predict nuclear proteins. And all of this is combined in a machine learning device. Sort of the later, and I misspelled here, the acronym is written whatever. W, capital O, capital P, capital S, and everything else is small. That's Wolf P-sort. Is again, this one grinds up a protein into features that are relevant to predict subcellular localization, and then uses k-nearest neighbor and Bayesian inference for that. That's the current best method out there from the P-sort series. Other methods include Sherlock. Sherlock is the idea to use text mining information. So if in the text mining you find evidence for the subcellular localization in a paper, let that help you to classify that protein. And that is the early days here. This is still relatively simple matching of words. So the you find localization in the abstract. Here, that is the idea. Then you find that this protein is nuclear, end of story. Here, you try a little bit more by also using sequence-based prediction, and by in fact refining the text mining, where you're not just looking for the keyword, but the keyword in a context. The refinement is one where you in fact now look at SVMs that look at the target signal. There are features that have to do with evolutionary conservation. There are motifs that could be in NLS and the like. There is something that has to do with the Go number, so the location in the Go number of the compartment, the CC hierarchy. If you have that information, look it up. Put all of that into a protein profile vector, and then do a one against many classification, one versus one classification of SVM that is somehow similar to what I showed you before, where the difference is that they, in this initial approach, simply picked the highest, the one with the highest performance, the highest prediction or the highest value to be the winner. Multilog is essentially something like that, but it expands much on this SVM before the text analysis part. It is, you see some element here for the SVM target, you see the motif search, but it expands on the way features are pre-digested and before they are put into an SVM. In some sense, you can think about this like deep learning for SVMs. So you have sort of pre-processed features. You have several levels at which you sort of process the features and put them into new devices. The difference, again, to the deep learning idea that we typically see today is not trained as one device, it's trained as separate devices, which has advantages and disadvantages. It comes from a group in Tübingen, from Olga Kohlbacher. I'm not sure why. So this is another Multilog 2. In fact, adds the Go classifications that I showed you before, but it also adds really information from alignments. Multilog 2. Cello. Now we have a method that is not performing very well, but introduced a new concept in terms of the way it codes the input. So it begins by having a special composition for the N-terminus. You have already proposed that, is trivial. But then it partitions the sequence into k-mers, and then actually not k-mers where you look at the sequence, but k-mers where you look at the composition. Okay? So it's a sliding window approach with a running average of our amino acid composition, which nobody ever else tried to do. And I believe this is just important to remember in your head. I've never seen anybody use it in a way that it makes a difference, but this so far is the only way I have seen this. And that, I don't know what the reason is why this didn't work so better than other things. There can be many reasons, but I believe the idea is an interesting one. Maybe you encounter a problem where this could actually work. So you could go into more detail, and that is briefly, I mentioned already, work that is done by Michael Boden, who will possibly give a lecture in January on his latest work. He got big on developing Bayesian networks, a Bayesian inference-based motif finder meme. So has published some papers, some of them very visible. Nevertheless, the H-index is still low, but does very interesting and cool stuff in machine learning. One thing that he tried was, I talked about the nuclear localization signal that decides that a protein is put into the nucleus, but the nucleus has subcompartments, and well, they're actually not shown here. I should have put a slide. So there are different subnuclear compartments, and Mikhail tried to in fact distinguish these subnuclear compartments by having a different model for the nuclear import. So having a model that has different types of tries. Essentially, I said the nuclear localization signals are so diverse that you cannot put them into a machine learning device. Now, this is the first way to have a very complicated Bayesian model that would somehow learn a generic feature in a nuclear localization signal and would use that as one part to get into a device that classifies and possibly can do more than nuclear or not nuclear. So this part here is the part where they say that they can identify the nuclear localization signals on their own. And then comes the Bayesian network, where we look information from protein interactions, from how compact a protein is, from something such as this nuclear localization signal, to infer some subnuclear compartment. And you see chromatin, the nuclear pore, speckle, PML body. So some of them you may have seen before, nuclear speckle. So it tries to, in fact, classify proteins into these subnuclear compartments. Now, when I talk about compartment, the word is beginning to not make sense in this subnuclear classification, because they are no longer membrane enwrapped. So these are identifiable substructures in the nucleus. Identifiable means that some proteins are always part of the speckle. And you can see that. But the speckle, the way to think about the speckle, and that's also why the protein interaction was so important for that on the previous slide, the speckle is defined as an aggregate of proteins interacting. So imagine it like a large machinery of things that sit together. Like a ribosome or something. Again, a ribosome is misleading because the ribosome is a compartment, is membrane enclosed, partially at least. So it's a large machinery, and that large machinery has a reliable existence. Let me call it this way. The definition of a reliable existence, in every single cell, it will be there for the duration of the lifetime of that cell. Okay, that's one way to call it a reliable existence. So now people begin to see that some developmental signals, some function relates to these aggregates that are temporal. So some proteins come together, and whether they're together or not, it influences whether that particular plant, so it's research done in Weinstefan, whether that particular plant is easier to recover from a drought. And those, again, are subnuclear arrangements, or extracellular, partially extracellular matrices arrangement of things that exist only for some time. Not for the lifetime of that plant, or not for the lifetime of a cell. Okay? Again, so there is some level of accuracy shown here, the area under the curve. Yes?
If we start talking about subnuclear compartments and even smaller compartments, at what point would it make sense to start saying there are proteins that are transported all over the place, and there is no right way to predict whether or not they're in what?
But again, this is part of the exercise, right? One of the questions in the exercise is the data supporting the idea that proteins are located in two localizations? And then ultimately, in order to address that question, you have to say how much information do you gain by calling it 2, or how much of random do you add? And this would be true for what you say. So if you completely open the can to n states, then again you ideally would evolve an information content measure that would tell you that that the new state you introduce here is one that is statistically significantly observable. Okay? And there's a bunch of ways of defining that. One way is you do the experiment many times and you have an agreement on that number of states. If you don't, well, it could be because you cannot observe it because it's too temporal. But then I would argue that there's a limited value in trying that, right? So when do we lose that? And when do we still have something that's clearly, as long as we have membrane around, it's easy. Here, the story begins to be complicated.
Is there anything out of biochemistry that would hint at what the true distribution is?
No. So that brings us back to the story of how many membrane proteins are in human, how many enzymes do we have. So no. So biochemists typically do not have any relevant evidence for that. In fact, it's the other way around. So in this paper that we are writing up currently to show this error correction, we also, in the context of that work, we also showed that using the experimental data as a proxy for the truth is getting even worse, because the experimental methods have a clear bias for some classes. Some proteins they just can't get, for instance, membrane proteins. And that, in fact, leads to a situation where you sort of take the experimental data that you have, the incomplete experimental data, and you do worse than when you take the erroneous predicted data. One has more errors, the errors can partially correct for, but does not have the bias. And one has the bias, is more accurate, but has the bias. Anyway, so there is an issue here. And what do you immediately jump at? Is there something that immediately jumps at you?
Nuclear lamina.
Why?
Because I like the number 77.
Okay, fair enough. Any other comment?
Well, the speckle and maybe the pore, I don't know how it's defined really in the context of the paper, but since they're transient and like defined by protein interaction, it's like you only use the information you have more or less to say, well, those interact together, so they must be in the speckle. And it's not a real prediction, I guess.
Well. I mean, this is...
Well,
it is.
If your machine learns an interaction and it just combines which interact with mother, it's not an interaction prediction, is it? So it only uses already established interactions.
That is true, but it has to decide which ones it takes and which ones it doesn't. It's not calling everything a speckle that is interacting. So while you are right, there is in the Bayesian inference or Bayesian network, there is a decision to make. Some we group and some we don't. Yes, that's true. I see where your concern is, but I'm not completely sharing your concern to the full length of it. Ultimately, you end up with a decision, and that is what I call a prediction. And that decision is you take known data and you do something new with it. You could not have looked at the data and said the same thing. You may have looked, so when you see the speckle, you may have said, okay, there's nothing new because they're all interacting. But then there are others that are also interacting and they're not called speckle. And that's the decision. That's the classification. That's why it's a prediction. But it's not what I meant. Just look at the AUC and the numbers.
Well,
yeah?
one thing that pops out is that for the perinucleolar and the nuclear pore.
Yes, that's one way to in fact start. Yeah.
So we have like the smallest number of proteins.
Yes.
In both cases, but the highest accuracy.
Yes, that's exactly the point I meant. And this is something that will happen again and again. So you will observe that where the number is so small that in fact you just coincidentally may not have a variety in there, and you essentially get the same things again, and that's why you get such a consistent data set, because it's so small. If somebody had a new experimental method and you could get suddenly 100 proteins here, you would see that this is a complete overestimate. So the interesting observation, and this is not machine learn specific, this is also this happens when you do simple data processing, data analysis, or whatever you call that. You see that over and over again. The smaller your data set, the more you're inclined to overestimate your performance. So small data set, be very, very careful. That is an important issue here, and you see it here. Now the problem is, this is all we can do. We can possibly say, okay, there are some numbers here that we don't trust. And now we still don't know, is that because these compartments are not well defined? Or is this just because I don't have enough data? So, yeah?
Well, I would say if you take out, for instance, the highest values of accuracy and the lowest, and then you take in...
No, but wait. So this number 71 essentially is biased by where most of the data sits.
Yeah, but what I meant was, when we look at the chiral body,
Yeah.
it has the same amount of proteins as the nuclear pore,
Yeah.
but it is nearer to the accuracy of everything else.
Yeah.
So that is like a dead giveaway that something might be wrong with the nuclear pore.
No, but that could. So again, Benjamin, I'm attacking the logic. It could be because it's more difficult to classify that. So let's get back to the SVM hierarchy we had before. We began with the premise that some classes are better to do than others. That was the original classification that you suggested that we in fact used. I believe Tatjana's story is true. It's always true. There's always some proteins or some classes that you get better than others. So that could be the reason here. But what is really your first observation is really the crucial one. So the highest number comes for the lowest count, the second highest for the second lowest count, so something is clearly going wrong. Clearly we overestimate for small classes. Again, when I'm at this point here, I can in my paper say that. And I can say there are things that I don't trust. I could possibly put them in by increasing the standard deviations. So yes, in fact, these standard deviations are the highest. I suspect that they may still not be as high as they really are. So essentially what I assume is that they did the statistically correct thing of estimating standard deviation, but that tends to in fact underestimate your error, in particular in small classes. So most likely this is, so all you can do is say, I'm not going to publish the method that predicts those. But on the other hand, you know, if somebody is interested exactly in the nuclear pore, so this again is important for various drug-related issues. Nuclear pore is an important part. So many people may possibly want the method exactly for that. Yes.
And maybe it's also wrong to not publish failure because we all can learn from failure, and also...
Well, it's not quite failure. Be careful.
No, I did not say that it was your failure. I just said, you said if you have unsatisfactory accuracy results, you'd be like you'd rather not publish them. Yes,
I tend to do that.
My counter argument would be, well,
we can discuss that offline. I have something to say for that. We can discuss that offline. But all I'm trying to say is, once you have this data, ultimately all you can do is make a flag, be careful. But I don't actually know how to handle that, and you can't tell the user, other than being careful, I believe it's not right to say, never use it for that prediction, for instance. Or it's also not, let's begin from the extreme end that you just said, Benjamin. It's certainly also not okay to just not publish at all, because there are some numbers that are too small. Or to leave them out, most likely is also not right. So ultimately, all you can do is have this table, show this table, and flag issues in the discussion. Okay, and then you can now get to the point of, in fact, predicting new proteins in human that you didn't know before. And this is one of those predictions. And here in the perinuclear, you just find in the entire human one. So there is some issue with that. But the other one that was small, 12, now this is 12 is a set to start to look at experimentally. So it is clearly somehow useful. Now, here is an idea where we use subcellular localization to go to another level. The method initially was called Peanut, and I want to just show the idea. The idea is you have a bunch of proteins, you get sequences for these proteins, and then you search databases of protein interactions. Essentially, you identify all the interactions that are known, and then you label the protein. You label the protein for each interaction between a pair of proteins. You look up the subcellular localization, you look up transmembrane helices, and you look up the Go classification. And then you ask, how often do proteins interact that come from two different subcellular localizations? Now, in this particular case here, green means more than expected, red means much less than expected, and white means essentially as expected by random, right? You see that the diagonal essentially is green, and that there are some exceptions to the diagonal. Now you can sort of take this level of color here and project it into a probability. And that then allows you to re-annotate a pair of proteins and to say, well, some of these pairs of interactions are very likely and some of them are unlikely. And you can also take these interactions and put them onto an image of a cell. So we're now, instead of just having the cloud of interacting proteins, you have the cloud of interacting proteins in compartments and sometimes spanning over different compartments, right? And this is in fact another piece that is currently has been just done. Now it's called CellMap by Christian Dalago in our group. So initially this was done by Yanai Ofran. And when Yanai first published this in 2006, he identified, for the Alzheimer pathway, he identified one particular protein, Q9P2H0. And that protein was here. Now what you see is, sorry for the quality of this slide, but you see there's a bunch of proteins here, there's a bunch of proteins here, and then you have a protein that has no identifier that you can recognize. So all you can see from this identifier is a uniprot identifier of a protein for which most likely you don't know anything. Otherwise it would have a Swissprot identifier. Otherwise it would have a name of something that characterizes in the name, essentially is supposed to characterize what it does. So this is one that you don't know anything about. So if you looked at the classification of these pair interactions, the first one you would never look at is this, because you know nothing about it. When you look at a bunch of proteins that interact with a protein that you're interested in, you look at things that you can look up some biology for, and then you hope that you can design your next experiment. But you would never look at things where nothing is known. Putting it onto the subcellular localization, not adding anything else, just putting visually onto the subcellular localization, you see that this is the one that goes through lipids, through vesicles. So this is it's the only connection to the secretory or to the outside, to the extracellular space through vesicles. So this puts an importance on this particular protein. Okay, and in fact it turned out to be a protein, so somebody picked this up experimentally and it turned out to be an important protein. So this is one of those examples where you can just use visualization to discover something that immediately could be interesting. In this particular case, the one thing Yanai tried did work. But he has a lucky hand. And again, so we now have somebody who has put something like that into a more modern JavaScript framework. Briefly to the systems, and I believe I'm going to stop here and just leave it to questions.